This is the smooth muscle lecture. We're going to look at the differences between skeletal muscle and smooth muscle. So, anatomy review first. Remember that a smooth muscle cell is a spindle-shaped cell in that it is fat in the middle and narrows to a point on each end. There's only one nucleus and there are no sarcomeres. Because there are no sarcomeres, we have to have a different arrangement of the actin and myosin. So what we see is that the actin is not attached to Z lines. It's attached to the membrane. It's something called a dense body. And when we see that the myosin pulls on the actin to bunch it up, instead of having a nice end-to-end smooth contraction where we just shorten the cell, the muscle tends to bunch up kind of like a pillow that's all been squished towards the center, because we're going to pull the sides in as well. And this is a photograph or a drawing I took from your textbook where we have the dense bodies here on the membrane. And you can see how the actin is attached there. And it shows these actin with the myosin here in between the actin. And when we pull it as a bunch up. So instead of having it be just the ends coming towards the center, you can see we're also pulling the middle through. So it's kind of a pull in towards the center, pulling those dense bodies closer together. And the other thing that's very different about smooth muscle is smooth muscle doesn't have troponin or tropomyosin. So instead, we have to think about how smooth muscle is different. Now, there are different subtypes, but the key here is that calcium comes from the extracellular fluid. Smooth muscle actually has very little sarcoplasmic reticulum. It has some, but it has very little. So most of the calcium is coming from the extracellular fluid. And these calcium channels are open in response to hormones and neurotransmitters that work through the G proteins. We don't get a ligand-gated channel. Instead, we get the neurotransmitter activates the receptor is an enzyme that activates the G protein activates the secondary enzyme that then produces a second messenger. The second messenger then acts to open the calcium channel. It's longer. It takes more time. Smooth muscle is much slower. And depending on the location, the sympathetic and parasympathetic, so when we say nervous system, we're talking ANS. We're not talking SE, we're talking VE. So it's autonomic nervous system, so sympathetic, parasympathetic, and we might have both innervations coming to smooth muscle. Skeletal muscle gets one neuron. Smooth muscle might get the pair each side, not always, and sometimes hormones that circulate through the body. Remember the hormone lecture where hormones go through the blood, and they can control the amount of calcium in some smooth muscle. And we have two types. We have single unit smooth muscle, all the cells work together like a single unit, and we have multi-unit, which the cells work independently. So we will come back to those, but those are the types. Let's talk about smooth muscle contraction mechanism. So I already said that we do not have troponin tropomyosin. Instead, we have covalent regulation. Remember that troponin and tropomyosin are all allosteric, which we saw in skeletal, and we will see again in the heart. Instead, we get this covalent regulation of the myosin. Remember, overall, we have to activate the ATPase activity. So here, calcium comes in from the extracellular. It attaches to the protein called calmodulin, which stands for calcium modified protein or calcium modification protein, depending on who's writing the book. But calmodulin is then allosterically activated by the calcium. So step one, allosteric. Then when calmodulin is active, it's going to allosterically activate a protein called myosin light chain kinase. And the key word here is kinase. We already know that kinases cause covalent activation. So this enzyme phosphorylates, we add our PI, just like before, to the myosin chain, which covalently activates it so that it can actually burn the ATP and cause movement. When we talk about the time difference from the action potential to starting contraction, smooth muscle is about 100 times or more slower than skeletal muscle. It's about 10 times slower than your heart or more. So I pulled this book from, this picture from the book showing where the calcium is on the outer membrane. And I drew camp here just saying in some cells, it is the second messenger cyclic AMP that opens these calcium channels there. And in other cells, we use IP3, which is a different second messenger. So this is what I meant earlier when I said second messengers open the channels. So then we have calcium attaches to calmodulin, which then activates the kinase. MLCK, remember, means kinase on the end. We get this phosphorylated thing. We activate the ATPase, and this is covalent. And we get contraction. If we don't have that, it's not working, and we get no movement. So think about the contraction mechanism. Once the myosin has been covalently active, the myosin can attach to actin. We don't have to control the actin this time. We control the myosin this time. And we always still need ATP in order to release and keep moving. ATP is that energy molecule. But the key here is that without this activation, myosin can't actually attach to actin. Relaxation in smooth muscle is also more complicated than in skeletal. It has at least 2 steps. First, we have to pump the calcium back out of the cell, whether we pump it straight there or we pump it into the sarcoplasmic reticulum. It's still pump. This means calcium is being moved up its gradient. It comes off the calmodulin. That was only allosteric. That's easy enough to do. But the second step is another longer phase. We have a phosphatase. There's that ACE ending. Because even if we deactivate the calmodulin, the myosin is still going until we take off the covalent activation. So what we need is the other enzyme to deactivate the myosin, right? So this is a double process, which means it's going to take even longer. It can take several seconds. In some of our data sets, it will take 8 to 15 seconds. When we think about the difference, smooth muscle might only beat 4 to 6 times a minute.